We will get started, so we don't come up on time. Brief introduction, of course. My name is Dennis Koester. I'm your host here, and we're today talking about the 2023 drug approval highlights. I'm here with our CEO Dennis Hu and with our director of scientific research Lou Pennington. Hi Lou and hi Dennis. Good morning. How are you guys doing?
Morning.
Good morning. Doing well.
So the drug approval highlights that we're going to be talking about today are actually all pulled from the Drug Hunter webpage, and I briefly want to show you one of the articles here that we have a highlight about. So this is ritlecitinib. Lou is going to be talking about this in detail. You can find hundreds of these case studies on Drug Hunter. We talk about what is really interesting about the project. We talk about what was the starting point for the discovery, any challenges that were overcome in the lead optimization campaign. We're talking about binding. We're talking about pharmacology, about the synthesis as well, and about clinical differentiation. So a lot of interesting depth here in all of these articles. And again, we have over 600 of these articles on the website that you can be learning from. And with that, I'd hand it over to Dennis Hu to start our presentation. And we're going to start with little highlights on approved drugs and then go into 4 case studies that we have selected as really exciting that we'll be sharing with you guys. So super excited for this session. Dennis, go ahead.
All right, thanks, Dennis. Well, shout out to Dennis for putting these slides together. I think this ended up really nice and made it really easy for me and Lou. So all credit goes to Dennis for this presentation.
Thank you, Dennis.
Yes. So what we'll do is we'll quickly scan through a bunch of these approvals and give you some fast facts. There's a ton to talk about, and as Dennis mentioned, we have more in-depth articles on almost all of these molecules on Drug Hunter. So if you want to learn more, you can go there. But I thought we'd leave you with something quick to know about a fun fact about each of these drugs. So pirtobrutinib on the top left, it's the first reversible BTK inhibitor that was approved. This molecule has an interesting story. It was actually discovered by a British biotech, Redx Pharma. Unfortunately, Redx had run into some troubles, I think, and sold this molecule or out-licensed this molecule to Loxo for around $40 million in 2017, I think. Loxo, a few years later, was acquired by Lilly for several $billion, not just for this molecule, but also for some other kinase inhibitors. But it's a really remarkable compound because I think people assumed that a reversible molecule would have to be larger or need to be bigger to access selectivity pocket. But this thing seems to be doing quite well without that. It also has 5 hydrogen bond donors, yet is an oral molecule. So that's something notable about this as well. Second molecule, elacestrant, is an oral selective estrogen receptor degrader or modulator. This molecule is also pretty interesting. It was discovered by Eisai, I believe, before being out-licensed and developed by Stemline, and most recently, I believe Stemline was acquired by Menarini or partnered with Menarini to distribute this globally. Elacestrant is interesting, and I believe it's the first new SERD approved in quite a while. It's the first oral SERD, I think, that's been approved. There is an injectable one, fulvestrant, that's been used for quite a while, or intramuscular one that's been used for quite a while. But there's been a big race for oral SERDs to help more breast cancer patients. There are several oral SERDs in late-stage clinical development, and this molecule is interesting because it not only can be administered orally, but it also seems to have a broader effect on estrogen receptor mutants. So hopefully this is a sign of good progress to come in breast cancer. Third molecule, quizartinib, is a rare approval for acute myeloid leukemia. I think there was almost a 2-decade gap before a few recent drugs were approved for acute myeloid leukemia. So it's good to see a couple more molecules being approved in this space. There are a few FLT3 inhibitors that are approved. This one is interesting because of its unique structure. That heterocycle in the middle there is quite interesting. We looked this up in our drugs database, and there really isn't anything else approved that's too similar. So it's an interesting motif that is now a bit more validated. On the right is momelotinib. This is a JAK inhibitor that was discovered by an Australian biotech, Cytopia. Actually, Cytopia was co-founded by the discoverer of the JAK enzyme family, Andrew Wilkes. Andrew was a guest on a webinar a couple months ago, or maybe a year ago now, where he talked about the discovery of the JAK enzyme family and eventually the discovery of this molecule. This molecule has a fascinating story, and I encourage everybody to watch the webinar and read the articles on Drug Hunter about this. Even though it's a simple-looking kinase inhibitor, there's a lot to this, both commercially and scientifically. From a scientific perspective, what was interesting about this was one of the key differentiators about this molecule in myelofibrosis is a better side effect profile, which is hypothesized to come from an unexpected off-target, ACVR1, which is sometimes called ALK2. And so co-inhibition of ALK2 creates a better overall tolerability profile than JAK inhibition alone. Commercially, this is interesting because Cytopia actually had a partnership with Gilead, I believe, and Gilead outlicensed this molecule for about $5 million a few years ago to Sierra Oncology. Sierra Oncology took on development, got phase 3 data, and was sold to GSK for over $1 billion. Nirogacestat on the bottom left is a gamma secretase inhibitor. This one is interesting because it's a gamma secretase. It's the first approved gamma secretase inhibitor. And as many folks know, gamma secretase was a hot target, not in oncology, but in Alzheimer's for a long time. So it's fascinating to see this approval, not in CNS indications, but in desmoid tumors, a rare type of tumor. That's interesting as well, because one of the issues that people ran into with gamma secretase is inhibition of gamma secretase, affects A beta, but it also affects another pathway, Notch signaling, which is important for cell proliferation and survival. And that led to some undesired off-target or on-target effects that people didn't like to see for Alzheimer's, but turns out to be quite good for treating some of these rare cancers. So this was a Pfizer molecule that was outlicensed to SpringWorks, which has taken it through to approval. So very interesting story. Repotrectinib, we'll save for later because I think we will do a longer discussion on that. But it's a nice little macrocycle targeting ROS1. Fruquintinib on the bottom right is interesting as a VEGF receptor inhibitor. What I think is notable about this molecule is it's a rare recent example of a benzofuran containing drug. However, when you have a dose as low as 5 mg orally once daily, it looks like you can do well with anything. Excuse me. On the bottom right, we have the first, we have the first approved AKT inhibitor. AKT is a kinase upstream of the PI3K family of kinases. There have been a lot of efforts on AKT, and this is the first approval in that space. And this is a program that originated in a partnership between the ICR in the UK with AstraZeneca. So it does look like there are a handful of AKT molecules still in development. One of the concerns had always been therapeutic index with these sorts of molecules. But people are hoping that combinations of this with different things will show some better activity in the future. All right, so there are quite a few interesting molecules for rare diseases as well. The first at the top left is sparsentan. This is notable for a number of reasons. One is the use of a surrogate biomarker to achieve approval. So one of the challenges in renal drug discovery has always been, in many of these diseases, especially for chronic kidney diseases, you had to have very long clinical trials to look at overall survival or long-term benefit. However, more recently, the FDA has started to accept surrogate biomarkers like proteinuria or protein levels in the blood as surrogates to look at kidney health. And sparsentan is one of these recent drugs that's been approved based on changes in blood biomarker levels rather than long-term kidney health. This is an interesting history. So this is an old BMS drug from many years ago that actually Martin Shkreli's company Travere took forward all the way through to approval. So many people don't realize this, but Martin Shkreli, even though he was sort of notorious for another molecule, Daraprim, the drug price hikes, etc. Actually, where he made, according to him on a recent podcast, where he made most of his money was on the advancement of this program under Travere Therapeutics. So Martin Shkreli has an approved drug. All right. The second one here is omaveloxolone. I won't talk too much about this one because I think Dennis Koester is going to talk more about this, right? Okay. Trofinetide. I had to look at this one, Dennis, when I looked at the dosing, 5, 6, 8, 10, or 12 grams orally BID. That's not a typo, right? Is that grams?
I would think so. That seems like a really high dose, though, almost like an antibiotic. I'll double check that one.
Sure. But super interesting peptide. It's a GP tripeptide for rare disease. On the right, we have zilucoplan, which is a complement inhibitor. It binds to C5 and inhibits its cleavage. This is a injected or sub-Q molecule for a rare disease, myasthenia gravis. And this is interesting because it is derived from mRNA display screening. I think a portion of this macrocyclic peptide was derived from mRNA display. And this is the first approved peptide derived, or macrocyclic peptide derived from that technology. So this was from Ra Pharmaceuticals, a Boston area biotech that was ultimately acquired by UCB. And it's the same company that partnered with Merck on their molecule of the year MK0616's starting point from mRNA display. On the bottom left is leniolisib, a PI3K delta inhibitor. This one is interesting because it's a rare example of a PI3K inhibitor whose first approval was outside of oncology in this rare disease, APDS. It's originated at Novartis and was out-licensed to Pharming, which took it through development. And it's a nice, beautiful-looking little molecule that's a kinase inhibitor, but has some more sp3 character in its core and is a bit more polar and smaller than you might expect. The second molecule on the bottom there, palavarotene, is a RAR gamma agonist. This is also for a rare disease, and this is a long history from Syntex and Roche, and was more recently developed by Ipsen. RAR gamma and related transcription factors have been hot targets for a long time, but it's been difficult to find the right indications for these molecules. This is a nice example of a approved stilbene containing drug that, again, when you're at 5 mg orally once daily, you can be successful with quite a lot. On the bottom right are iptacopan, which was a molecule of the year candidate, I believe, in 2021, long before it was approved. That molecule is interesting because these proteases are notoriously challenging to drug. Novartis had an extensive hit-finding campaign to find starting points for this. I think they threw everything at this, and there's a fantastic story about this. A small anecdote here, I believe Nello Mainolfi was the project leader for this program at Novartis, and many folks may know him from Chimera of targeted protein degradation fame. So a nice example of a CEO who was once a chemist. On the bottom right, well, he's still a chemist, but it's great to see that history. Bottom right is vamorolone. It's a glucocorticoid receptor agonist for Duchenne muscular dystrophy. Another interesting example of a steroid drug with a interesting I don't know what we even call that, a semiquinone or something like that on the bottom left. Okay, so with that, I'll hand this over to Dennis.
Yeah, thank you so much, Dennis. Super interesting facts here. Yeah, let me cover the infectious disease approvals in 2023. So there was an antibiotic that was approved. That's a beta-lactam in combination with a beta-lactamase inhibitor here, sulbactam and durlobactam. And this is for really hospital-acquired bacterial pneumonia. And it's for a particularly nasty gram-negative bacterial strain. ABC is the abbreviation for that. So, this is really meeting an unmet medical need here for carbapenem-resistant gram-negative bacterial infection. Second molecule here, a combination of taurolidine and heparin. Interesting use case here in catheter-related bloodstream infections in patients with kidney failure. And here, interesting about this particular molecule is the delivery method. So this is for patients, again, that experience kidney failure and that already receive hemodialysis. And this treatment here is actually delivered via a central venous catheter. So kind of really interesting, innovative delivery mechanism. Then, of course, an antiviral that we're all familiar with, Paxlovid, has been approved, of course, for the treatment of COVID from Pfizer. Very inspiring story here, of course. It's a combination of nirmatrelvir, which is the Mpro inhibitor, and ritonavir, which is an HIV protease inhibitor, but is used as a CYP inhibitor here to extend the half-life of nirmatrelvir. And the really interesting story about this is this program probably set a record that can't be broken for the speed of first synthesis to first kind of approval. So this molecule was synthesized first in July of 2020, and the FDA gave it an emergency use authorization in December 2021, which is 17 months. So now in 2023, this was fully approved, and the speed of this program inspired many, many other drug discovery programs, even beyond infectious diseases, to speed up the development process. So super, super inspiring story. Then on the right-hand side in the top line here, risafungin, an antifungal, also really interesting peptidic macrocycle. As Dennis mentioned for zilucoplan, mRNA display screening is on the rise. This compound is not from mRNA display screening, also is delivered IV here. As Dennis mentioned, we are now seeing orally delivered compounds, macrocyclic peptides from mRNA display screening. So what's interesting about this compound is it was optimized to have a better metabolism profile, in thereby it could have an extended half-life, and the reactive metabolites could be reduced so that there'd be a better safety margin. Really interesting story here as well. In the OB-GYN space, we have Vizoa here, which is a NK3 receptor antagonist, and is used for hot flashes in menopausal women. So I think this is just a very nice example for a first-in-class drug. And Wendy Young gave a beautiful talk at the ACS in New Orleans about women's health, and we have a nice article on the website as well, highlighting why women's health is so important. And I encourage everybody to read about that. In the dermatology space, we have ritlecitinib, which Lou is going to cover in detail, so I'm not going to steal his thunder. In endocrinology, we have an SGLT2 inhibitor here approved for type 2 diabetes mellitus. And this molecule, bexigliflozin, is kind of interesting. Now, you may wonder, this is certainly not a first-in-class SGLT2 inhibitor. I think the first SGLT2 inhibitors have been approved more than a decade ago. But what is interesting about this molecule, it was optimized for its selectivity of SGLT2 over SGLT1. Now, why is this important? So SGLT2, let's just remember what this is doing. So SGLT2 is catalyzing the renal reabsorption of glucose into the bloodstream. So by inhibiting SGLT2, you reduce the reabsorption, the renal reabsorption of glucose into the bloodstream, and thereby lower the blood glucose level, which is beneficial in diabetes. Now, SGLT2 is primarily expressed in the kidney, whereas SGLT1 is more systemically expressed. So this compound here, which is a C glycoside, also really interesting structurally, of course, within the class, is actually 2,000-fold selective over SGLT1. So that has safety benefits. Now, here on the right-hand side in cardiology, we can see why this is important, because sotagliflozin is a dual inhibitor of SGLT1 and SGLT2. And we can see that it's used for reducing the risk of cardiovascular death and a heart failure hospitalization, indicating that the pharmacology is mainly driven through SGLT1, and that is also expressed essentially in the heart tissue. Also really interesting hemiaminal structure here with a thioaminal essentially, and otherwise very structurally similar to besagliflozin. On the next slide, we have the approvals in hematology, also really interesting. Second generation CXCR4 inhibitor motixafortide is used for hematopoietic stem cell mobilization in patients with terrible blood cancers like multiple myeloma that cannot mobilize enough stem cells. And the interesting thing about this compound is it's a second generation compound to plerixafor. The mobilization speed is way faster, and the mobilization yield is also higher, so that these patients that suffer from these terrible cancers don't have to undergo multiple apheresis sessions. It's actually co-dosed, it's dosed subcutaneously, and it's co-dosed with GCSF. Really interesting story here as well, from a compound that basically was delivered through academia into a collaboration with industry. Then also very interesting, daprodustat, the first in class approval in the US, at least, first FDA approval for a HIF-PHD inhibitor. HIF-PHD inhibitors have been approved in other regions like China or Asia in general, and Europe as well. And what kind of is a common motive in those types of HIF-PHD inhibitors is this glycinamide that you can kind of see here. And this is because these compounds are all mimics for 2-oxoglutarate. So really polar type of enzyme, the PHD class that is mostly inhibited with 2-oxoglutarate mimetics. In the diagnostic space, we had the approval of a PET tracer for prostate cancer. In neurology, we had 3 really interesting approvals. So zavegepant here, really interesting, basically a first CGRP receptor antagonist that is delivered intranasally. Of course, there has been a lot of research going on in CGRP receptor antagonist, but this one has like a really interesting delivery method. It's delivered intranasally, and the benefit of that is that the onset, this is for treatment of migraine, is really rapid. So within 15 minutes, pain relief is already observed, and it's also a great option for patients who suffer from emesis when they have to take oral treatment. So really interesting therapy here as well. Another interesting compound is zuranolone, here approved for postpartum depression from Sage Therapeutics. We have an extensive article on the website about this. It was also tested for major depressive disorder, but the FDA did decide not to approve it. But again, first in class oral therapy for postpartum depression. So another important contribution here to women's health. There was a drug that was approved for major depressive disorder, which goes through a different type of mechanism. This is a selective 5-HT1A agonist here, gepirone. And yeah, the interesting thing about this is really this kind of structural motive of this IMiD that you see here that we're used to from protein degraders. On the next slide, and that would basically conclude the section, we have a really interesting approval in gastroenterology here for ulcerative colitis with, again, a very, very low dose that we're seeing here, 2 mg QD dosed orally. And this drug has a very interesting story. It was acquired by Pfizer from Arena for about $7 billion, so really, really remarkable. And is a S1P receptor modulator. So that actually partially and reversibly reduces the lymphocyte count by blocking the lymphocyte trafficking at the site of inflammation in the colon. So really interesting MOA here. In the ophthalmology space, we witnessed 2 approvals. One is for dry eye disease. here, perfluorohexyloctane, probably set a record of number of fluorine atoms in an approved drug with 13 fluorine atoms here, and just reduces the evaporation of water in the eye. So really interesting type of modality here. Then last but not least, lotilaner, a drug approved from Novartis, and this is used as a first-in-class drug for highly prevalent eyelid disease, where 25 million patients in the US suffer from this disease. And what I found interesting about this is this kind of unusual motive here of this trifluoromethyl isoxazoline, I would say. So yeah, really something that you don't typically find in other drugs. And with that, I'll hand it over to Lou to give us a story about ritlecitinib. Lou.
All right, great. Thanks, Dennis. So as we were preparing to give the talk today, I told Dennis K and Dennis Hu that I've never actually worked on a kinase program before. I've been a medicinal chemist for 25 years and never had that pleasure. And I can guarantee you everything I know about ritlecitinib is what I've learned from the Drug Hunter article. So hopefully I can show folks that you can go from complete ignorance to actually knowing some pretty interesting facts about a drug discovery program. And this really is a cool story from both a biology and chemistry standpoint. It's a really nice testimony to the complexity of biology and how with enough patience and enough persistence, you can really interrogate that biology, those pathways and targets, and come up with therapeutic interventions for that. So ritlecitinib, what is it? It's a covalent kinase inhibitor that's intended for use in autoimmune conditions. You can see the nice structure of the molecule here. It's got a molecular weight of about 285 Daltons. And ultimately what has been determined is that this molecule is a dual inhibitor of both JAK3 and the TEC family of kinases. And this comes from Pfizer. And this was approved in June of last year for alopecia areata. So what is alopecia areata? Literally, this means patchy baldness. So it's different from male pattern baldness, like I suffer from. And it's caused by an autoimmune attack on hair follicles, leading to baldness. And about 2% of the general population is going to suffer from this disease in their lifetimes. So the previous standard of care for this disease condition were topical steroids or systemic immunosuppressants. And so the target rationale for going after JAK3 for this disease indication was that alopecia areata is driven by cytotoxic T cells signaling through the IL-15 STAT5 pathway. And as you know, we all know JAK1 and 2 inhibitors have shown efficacy, but there are a lot of safety issues associated with JAK1 and JAK2 inhibitors. And so to go after sort of a non-oncology condition and really try to attack this disease is safely and effectively as possible, the therapeutic hypothesis was that selective JAK3 inhibition might be beneficial for that. As JAK2 is involved in hematopoiesis and JAK1-dependent IL-6 signaling. And so one other nice thing is that JAK3, unlike JAK1, JAK2, and TYK2, is selectively expressed in bone marrow and lymphatic tissue, where the other JAK family members are ubiquitously expressed. So on the next slide, we can see that tofacitinib, this is sort of one of the first JAK1 inhibitors that was approved, I think the first. It was initially believed to be a JAK3 selective inhibitor and later found to actually be more potent on JAK1 versus JAK3, as well as having some JAK2 activity. And how did this happen? How did this pharmacological story unfold? Well, basically using a traditional ELISA assay with an adenosine triphosphate concentration right around the Michaelis constant for each one of these kinases, it appeared that this molecule was potent for JAK3. But using a more modern caliper nanofluidics assay, as you can see there in the third column from the left, what they found is that selectivity actually went down. And in fact, when they had more physiologically relevant concentrations of ATP on the order of about 1 millimolar in this caliper assay, they actually saw that the selectivity had flipped, and now the molecule is somewhat more selective for JAK1 versus JAK3. And this was recapitulated in the whole blood assay. So this was obviously an interesting journey of understanding of the pharmacology of tofacitinib. And so if we go to the next slide, what they started looking at is they were further pursuing this program, looking for other sorts of JAK inhibition profiles. They had an X-ray crystal structure of tofacitinib, and you can see there on the far left in this X-ray crystal structure that that key carbon-nitrogen sigma bond off the heteroaromatic ring system actually orients the amide away from serine 985. Now that serine 985 in JAK1 is actually a cysteine in JAK3. And so to optimize the selectivity of the molecule and to try to engage that cysteine 909, they simply clipped off the two methyl groups, the one on the piperidine ring and the methyl group on the nitrogen atom off the heteroaromatic ring system, and then swapped out that amide with an acrylamide moiety. And what they found was compound 4. We'll talk a little bit more about the compound of the profile of compound 4 in the next slide, but this compound actually had decent potency and selectivity for JAK3, but it suffered from some stability issues related to glutathione reaction in the presence of glutathione transferases. And so to get around that, what they did is they appended a methyl group, and this winds up being axial methyl group, excuse me, at the 2 position of the piperidine ring, and that got around some of those chemical stability issues associated with glutathione reactivity. So what you can see in this compound 4 in ritlecitinib is that key sigma bonds rotated 180 degrees, and you can see that it now is positioned to react with cysteine 909. And interestingly, this molecule has fairly low binding affinity, as we'll see shortly, to the kinase, but the oxygen atom of the acrylamide that you can sort of see on the right-hand most image is engaging in an intermolecular hydrogen bond with cysteine 909. It's also engaging in a hydrogen bonding interaction with a water molecule that's not shown, that's in between that pyrimidinal nitrogen atom and the oxygen atom. So a really nice way of activating that acrylamide warhead system. So if we go to the next slide. And so, one thing that people have to think about when they're contemplating going after covalent inhibitors is the protein resynthesis rate. And the idea a lot of times is, though, that by taking a covalent approach, one doesn't need to maybe dose every day or worry about, PK issues and so forth. But unlike a lot of proteins like RIP2 kinase, I believe, and BTK, which have very slow resynthesis rates, the JAKs have fairly high resynthesis rates on the order of 2 to 4 hours. I think JAK3 was found to have a resynthesis rate of about 3.5 hours. So it's fairly rapidly resynthesized. And so they were going to have a reasonable pharmacokinetic profile for the molecule, despite being a covalent inhibitor. So shown here are the sort of profiles for compound 4 and ritlecitinib. You can see both of them are JAK3 selective compounds. Both of them have good stability in human liver microsomes. But you can see their rat blood half-lives are very different. Ritlecitinib is a little bit higher, and that's because that methyl group I talked about on the piperidine ring of ritlecitinib seems to dial out some of the binding affinity for the GSTs that lead to that glutathione conjugation and that elimination mechanism. So ritlecitinib was also found to have fairly reasonable clearance and also good oral bioavailability across species. And so one thing they had to use in this program is that whole blood stability assay, which helped monitor stability. So if we go to the next slide. So yeah, I love the title of this slide. Why is this little molecule a big deal? And I think the images on the bottom left of the slide say it all. So there's a patient on the bottom left. This is a baseline sort of amount of baldness that this patient experienced at the beginning of the study. And after 24 weeks of a therapeutic intervention, you can see by week 24, this person seems to have what amounts to a full head of hair. So this is an incredible efficacy for this molecule that was also matched by very good safety. We don't have that information right here, but of course, this molecule has been shown to be very safe. And this molecule is also, it's selective for JAK3 and the TEC family kinase inhibitors. And so one interesting thing you can see here is that despite having weak binding to JAK3, that really high K-ACT value that derives from those interactions we talked about earlier lead to very rapid covalent conjugation to the protein and really limit that non-specific covalent binding. And this remarkable efficacy is in contrast to intolerability is in contrast to that for JAK1 and 2. And so you can see one interesting thing, if you compare JAK3 and BMX, so the profile of the molecule, you see the Ki for this molecule in BMX is much better than, it's about greater than 10-fold better than for JAK3. But because of that K-ACT and the resulting K-ACT-Ki ratio, the molecule actually is more selective for JAK3 than BMX, as you can see in the IC50 and the column that's second from the right. Both of these molecules at OC50, that's a concentration at which you achieve roughly 50% receptor binding. So they have very similar OC50s. If you go to the next slide, so the other cool thing about this molecule, and says a lot about how we can interrogate and understand biology with chemistry, is that this molecule actually was used to disprove the JAK1 dogma. The JAK1 dogma was that, in the JAK1, JAK3 signaling pathway, the JAK1 was more important than JAK3, and that you couldn't get the sort of robust pharmacological response by inhibiting JAK3 that you could get with JAK1. And I think this table says it all. If you look at the JAK3 selective molecule, ritlecitinib on the left, and the JAK1 selective molecule, 554 on the right, you can see that ritlecitinib only has activity through the IL-15 pathway, looking at STAT5 production. Whereas the JAK1 compound actually has activity in that pathway, as well as the interferon pathway shown there, IFN, G, and A. So this molecule really helped understand the biology of this pathway and these therapeutic targets. So if we go to the next slide. So I'll just wrap things up. This is the synthesis of the molecule. So starting from 6-methylpyridine-3-amine, it was Bach protected using standard conditions. And then that pyridine ring was reduced to a piperidine ring using platinum catalysis. And then the nitrogen atom of that piperidine ring was protected as a CBZ group. The cis and trans stereoisomers and all enantiomers were separated using chiral SFC. I guess that's a racemic cis molecule there. That molecule was then deprotected, removed the Bach group. It was coupled to the aromatic system. The CBZ group was removed, and then finally the acrylamide was appended in the last step. So really a nice 8-step synthesis of this small molecule. Again, the molecular weight's about 285 Daltons. So really a truly small molecule with some really wonderful properties. All right, I think I'm going to hand it off to Dennis Koester now.
Yeah, thank you, Lou. Super interesting. So yeah, let's switch gears to omaveloxolone. So what is omaveloxolone? Omaveloxolone was actually discovered by Reata Pharma and Texas Biotech, and was then acquired by Biogen for about $7.3 billion after its approval. It's actually dosed 150 mg QD and is supposedly a nerve activator. It's an interesting story, actually, because essentially this is derived from a natural product, as we'll see, that's based in academia and was discovered about 30 years ago. So let's talk a little bit about Friedreich's ataxia. What's Friedreich's ataxia? That's actually a progressive neurodegenerative disorder, excuse me, that's caused by a mutation in the FGX gene, and that FGX gene is coding for a mitochondrial protein called fratraxin, and that fratraxin is responsible for the assembly of some iron-sulfur clusters in mitochondria. So in the pathology of Friedreich's ataxia, that it leads to an accumulation of iron in the mitochondria, and that leads to oxidative stress, neuroinflammation, and often shows in the loss of motor skills and ultimately in cardiac dysfunction. So omaveloxolone was the first approved treatment for Friedreich's ataxia, and it was basically approved on a phase 2 clinical trial with about 100 patients that showed a 2-year disease, 2-year delay of disease progression. And Reata actually got a rare pediatric priority voucher for speedy review because this is actually a program that is in a very rare disease. So at the first look of it, the FDA actually looked at it first in 2020 and did not want to approve it back then. And usually when those kind of things occur, there needs to be 2 confirmatory trials for efficacy, but the FDA actually had some regulatory flexibility, and with 3 years of confirmatory data and evidence that it was a nerve activator with the PD, approved this molecule here. So what is a nerve? What is the nerve keep axis? Let's talk a little bit about that, and let's talk about why this is something that is indicated in Friedreich's ataxia. So nerve is an interesting transcription factor that regulates the expression of over 250 genes. And it's regulated by, so KEAP is an E3 ligase that ubiquitinates nerve and readies it for proteasomal degradation. But when you inhibit KEAP covalently, you basically functionally activate nerve. And this has been made prominent by Biogen. Biogen discovered, Biogen discovered the MOA of DMF, dimethyl fumarate. Dimethyl fumarate is actually an antifungal that was also found to have anti-inflammatory properties. It was actually used in the 1950s by a German biochemist who treated his psoriasis with this. And in the 1990s, it was discovered that patients who suffered from psoriasis and also multiple sclerosis stabilized their disease with this, and was then found to be the first nerve activator. Biogen basically elucidated this mechanism in 2011 and received FDA approval for multiple sclerosis in 2013, and it has been really commercially successful. Now, DMF, as you can imagine, is not a very selective nerve activator and has actually some side effects, including leukopenia, which is attributed to HCA2R agonism, and also has GI intolerability issues. So Alkermes paired up with Biogen to develop a more GI tolerable drug. Now, as you can imagine, KEAP actually has multiple reactive cysteines, and omaveloxolone is a reversibly covalent inhibitor, as we will see. So actually, many natural products have been tried that have electrophilic warheads. And, again, nerve is kind of the master switch for oxidative stress and regulates, for example, the expression of GSH and GST. So, it could be indicated in multiple different diseases. So there are hundreds of clinical trials in different diseases going on at the moment. On the next slide, we will see the discovery story. So the discovery story of omaveloxolone is not actually disclosed, but the discovery story of a close analog, bardoloxolone, also called CDOO, is disclosed. And CDOO was found to be a nitric oxide production inhibitor and have similar properties to the anti-inflammatory dexamethasone in the 90s, and its nerve mechanism was only elucidated in 2005. So now omaveloxolone is actually the difluoromethyl propionamide of CDOO, so it's believed to bind very similarly. So as we can see here in this graphic, the cysteine of KEAP1, cysteine 151, binds reversibly covalently to this enone that we can see here. And that reversible covalent inhibition probably contributes to the long human half-life. And also what has been found too is with GSH, no stable adducts are formed, contributing to the plasma stability of this particular compound. So now again, KEAP basically is inhibited, and then NERV is not being ubiquitinated. So basically it's functionally activated. Really interesting kind of MOA here of NERV stabilization. On the next slide, we'll see a little bit about the SAR. Again, the discovery story is interesting and was started in the 90s with the discovery of a triterpenoid story and starting from the natural product oleanolic acid in a basically anti-inflammatory assay that was monitored with a cellular assay of nitric oxidase synthetase inhibition. The initial hit here was found to be this triterpene. And if you can see, this triterpene here was modulated in the C ring to feature this enone here. And that led to a 10-fold improvement in potency. And separately, if you introduced an alpha nitrile group here in that other enone in the A ring, that also led to a 10-fold improvement in potency in that particular assay. But combined, those two changes led to a 10,000-fold improvement and led ultimately to the discovery of bardoxolone here. Very interesting story here is actually that bardoxolone is not necessarily brain penetrant, and so it's an acid, and so is the methyl group. It was actually tested in chronic kidney disease. So basically, when Reata put this in a phase 1 clinical trial, they found that this had some benefit on the glomerular filtration rate, GFR, excuse me. So suggesting some benefit in chronic kidney disease. When they tried that in a phase 3 trial, they unfortunately found that there are cardiovascular complications, but they had some exclusion factors and drove this trial to completion in Japan. But unfortunately, it was found that it had effects on the biomarker, the eGFR, but did not lead to a delay in disease progression to end-stage kidney disease. So the development was terminated for chronic kidney disease in 2023. On the next slide, we will see the brain penetrance. So essentially, this is a really interesting example of a compound that is actually enriched in the brain. Typically, in terms of tissue distribution, the brain is the most impaired organ. But here we can see at all doses in the cynomolgus monkey study, PK study, the concentrations in the brain were actually 4-fold higher than the concentrations in the plasma, which is, of course, great for neurodegenerative diseases such as Friedreich's ataxia, and could also maybe help with other diseases like Alzheimer's and Parkinson's disease. Now, of course, we also see a little bit of an enrichment in the liver here, and the question was of if that is a concern, and we'll see this on the next slide. So initially in phase 1 studies, what was found is that ALT and AST were elevated without any signs of liver failure. So there was no sign of increased bilirubin, which would lead to a pathology called jaundice that is often seen in newborns, or albumin, a decreased albumin, but those were not seen. So it was actually known that this elevation of the AST and ALT levels was maybe an on-target effect, because NERV activation is known to increase the mRNA expression of those two transaminases. Now, transaminases are also expressed in different tissues, not only in the liver tissue, and was shown that in other tissues, it was also elevated and could actually help with the glucose metabolism. So the DILI committee of the FDA agreed with Reata's assessment that those elevations were benign. Now, omaveloxolone and other NERV inhibitors are now also tried in other indications. Now, this is the first time that a NERV activator, more selective NERV activator, has been shown to be efficacious in human. And it's seen now how this is looking like, the whole pharmacology of this in human. So this leads to more and more companies working in the space of the nerve keep axis. So for example, GSK, Astex, and Vividion are working on this. Vividion recently disclosed at the ACS their nerve 2 activator, and at the upcoming ACS in Denver, they will actually disclose in the first disclosures a keep activator. So that'll be a very interesting program. As I mentioned, KEAP1 is also an E3 ligase, and similar to HIF that we previously talked about, that led to a speed up in VHL as the use of ligase in protein degradation, maybe this program could lead to the acceleration of the investigation of KEAP as the E3 ligase in protein degradation. And on the last slide, briefly talking about the synthesis. So two interesting things here about the synthesis that was done. One is the fragmentation reaction that you can see here from this isoxazole to this basically to append this alpha nitrile group on the ketone was really elegantly installed with a fragmentation reaction that led to quantitative yield. And then the acid, the bardoxolone, was transferred into omaveloxolone with a very nice courteous rearrangement that took place. And with that, I'll hand it over to Dennis to talk about repotrectinib and pirtobrutinib.
All right, thank you. So down to our last two case studies. The first one here is on repotrectinib, which is helping to treat brain metastases in rare forms of non-small cell lung cancer. So repotrectinib is a kinase inhibitor that targets 3 kinases that are involved in different cancers. The main ones for lung cancer are ROS1 and ALK, but it also does target TRK. This is so ROS1 positive non-small cell lung cancers are rare, but and they lead to brain metastases in almost 50% of cases. ROS1 positive lung cancers are about 1 to 2% of all lung cancers. So it's an incidence that's similar to ALK positive lung cancers. But this mainly appears in younger non-smokers. So it's more, it can be quite catastrophic to be diagnosed with this mutation. Over 50% of patients develop resistance to previous ROS1 inhibitors, including crizotinib. And one of the challenges is that previous ROS1 inhibitors don't have very good intracranial activity, which is a challenge in dealing with brain metastases that are so frequent here. If you look across the older generations of kinase inhibitors that are approved for ROS1, you can see that they all suffer from different things. So the original crizotinib suffers from poor brain penetration. Larotrectinib, which is a TRK inhibitor, also has poor brain penetration. Entrectinib was later developed by Ignyta and acquired by Roche for over $1 billion. And a key component of that was the evidence of human intracranial activity with that molecule. And lorlatinib was also approved, which is an ALK and ROS1 inhibitor. It's brain penetrant and also has been wildly successful. The challenge with entrectinib and lorlatinib is they also encounter resistance. And so there's multiple properties that an ideal molecule would have, which is obviously selectivity, potency, brain penetration, oral bioavailability, as well as the ability to address many mutant forms of ROS1. So one of the great things about this small macrocycle chemotype is that it has a lower footprint in the binding site. And so by being smaller, more compact, and not relying on as big of a surface area in interacting with the protein, there are fewer ways that the target can develop resistance against this molecule. So shown here in the figure, you can see that crizotinib with that bulky piperidine that extends out to the solvent front, there are residues in the solvent front, like glycine 2032, that can mutate to bigger residues or charged residues like arginine that lead to a significant drop-off in activity. Whereas with the compact footprint of repotrectinib on the right, you can see that those sorts of solvent front mutations don't impact its binding as much. You can see at the bottom here that repotrectinib has a significant potency against that mutant as an example, the G2032R mutant, whereas crizotinib sees over 100-fold drop-off in activity with that mutation. And entrectinib as well on the right sees a 200-fold drop-off in activity. So being able to have a brain penetrant molecule that addresses resistance is great. That is part of why BMS bought Turning Point for over $4 billion to acquire repotrectinib, and next generation molecules continue to emerge. So Nuvalent is a Boston area biotech that has a very interesting next generation ROS1 molecule, NVL-520. There's a great article on this as well. And what's notable about that is people had thought before that TRK was an innocuous off-target before. There are molecules that specifically target TRK to treat different types of cancers. However, it's been found that actually TRK inhibition does lead to some neurological side effects. And so NVL-520 is designed to be more selective and only target ROS1 and spare TRK. So encourage everybody to read about that fascinating case study as well. Okay. The last case study we'll talk about is pirtobrutinib. This is the first approved non-covalent BTK inhibitor that shows a significantly improved safety profile in treating a range of cancers. BTK obviously is famously inhibited by covalent molecules like ibrutinib. But one of the challenges with covalent inhibitors, right, is they are heavily dependent on a single cysteine residue for compound alkylation. So obvious way that tumors can develop resistance is by losing that cysteine. So a reversible inhibitor can be attractive because they may be active in those mutations where patients lose efficacy with all of the other BTK inhibitors. These cysteine mutations, targeting cysteine 481, are in 80% of patients that have covalent BTK inhibitors being ineffective. And there are also tolerability challenges with some of the prior generations of covalent molecules. So if you look across the 4 generations of approved BTK inhibitors in the US, you can see that pirtobrutinib has quite strong activity against against a couple of cysteine 481 mutants. But importantly, if you look at the rates of grade 3 diarrhea, rash, and atrial fibrillation, which is a cardiac side effect, these are all lower than ibrutinib as well. And so it could differentiate based both on efficacy as well as tolerability. The reversible inhibition might also contribute to better PK, which also can contribute to resistance mitigation. So obviously with the covalent inhibitors, if you're dosing relatively infrequently and unable to cover the target for an extended period of time, it does create a window of resistance, even though those covalent molecules do deplete the active protein. The protein is resynthesized over time, and that does create a window where some of these mutants can develop if you're not nailing the target, so to speak. So with a reversible molecule with better PK, that does cover the IC90 in a sustained manner, that can contribute additionally to reducing the incidence of resistance. All right, so with that, we'll wrap up. There are numerous other case studies that go in depth into all of the other approved drugs on Drug Hunter. Again, really encourage people with an interest in these molecules to read more about these. There's so much more to the science and commercial and clinical landscapes of these molecules that we didn't have time to talk about, but are really fascinating and can inform a lot of other drug discovery programs. You can find all of these on drughunter.com. We also do have a full text and structure search for all of these articles. So if you're looking for certain concepts like hERG inhibition, PXR activation, or things like that, you can search across all the case studies for those sorts of topics. And there is an approved drug structure search on Drug Hunter as well, which you can use to look up, how many approved drugs have benzofurans or things like that. I certainly use that a lot in preparing for this presentation. So if you are a member and have access to that, don't forget to check that structure search out as well. All right, so thank you everybody for joining us. Thank you to our whole team for helping put this together. And we're happy to take any questions.
Awesome. Yeah, thank you. I'm realizing we're coming up on time, but we're still going. We have 2 questions already in the chat. So we have one question here from Gagan, wondering about the IP of momelotinib, whether this has been kind of covered before in a patent. So he's wondering about the novelty of momelotinib. Do you guys have any insight into that? Dennis, do you know the story best, right?
Yeah, so that's an old molecule, right? I mean, I think they
Yeah.
were working, they were one, Cytopia is an Australian startup that was, again, co-founded by the discoverer of the JAK enzyme over 2 decades ago. So they were one of the first people in the space, and they discovered that molecule through phenotypic screening. This was, again, 20 years ago, before people were doing broad kinase panels and had a lot more data. And that approach actually led to its unique profile, right? It's not just the JAK2 activity, it's also this co-inhibition of ALK2 that leads to its unique pharmacological profile.
Yeah.
So yes, as far as novelty, again, this was, given its relatively simple looking structure, this was patented a long time ago, before, I guess, the kinase landscape was, super saturated with a whole lot of chemical matter. But interesting to see that you can have a billion-dollar molecule with very unique properties with a relatively simple structure.
Yeah, makes
Just
total sense.
one generic comment on intellectual property. I don't know if folks remember Mark Merkel's 2018 perspective on what makes a great medicinal chemist. One attribute he said is not worrying too much about IP. If there's a will, there's a way.
Nice. Awesome. Next question is, can the omaveloxolone MOA be applied to other diseases associated with oxidative stress? I can answer that. The answer is absolutely yes. And people are looking at other inflammatory diseases, for example, lupus and arthritis, also cancer people are looking in. And it's, of course, you can do nerve activation, but also, keep activation, which would then lead to nerve inhibition. So it's currently looked at, again, by multiple different companies in multiple different diseases. The next question is, how will computational modeling enhance covalent inhibitor design? Maybe Lou or Dennis, do you want to take a first stab at that?
Just looking at the ritlecitinib story, I mean, this is a fantastic story about engineering an existing molecule to give it the activity you want in one protein, to give it the activity you want in another protein. And an understanding of the conformation was absolutely critical to that. If you remember, that carbon nitrogen sigma bond had to rotate 180 degrees in going from the original molecule to ritlecitinib. So yeah, it's gonna be 100% part of this. You need to know the conformation, you need to know the binding mode, you need to know the distance and angle of the warhead from the reactive cysteine, and then you need to think about how you're going to activate the warhead. That's something that has to happen. These things typically don't happen without some degree of pre-organization and in activation in the binding site. So it's absolutely going to be critical for that. And in fact, ritlecitinib, they did not have an X-ray structure. It was from modeling that they sort of interrogated all of this.
Yeah, no, and you can actually see this with a kind of also a kinetically selective molecule, lirafugratinib, which is a FGFR2 selective inhibitor that was discovered by extensive computation through kind of a supercomputer where Relay Therapeutics looked at the molecular dynamics of the different FGFR isoforms, FGFR1 and FGFR2. So yeah, computational modeling is certainly, in my opinion, going to enhance the discovery and enable the discovery of more covalent inhibitors. All right, I don't see any other questions in the Q&A. So with that, I want to thank Dennis and thank Lou for a very nice educational seminar. I learned a lot. I'm always learning a lot from you guys. Thank you to the audience for all the questions. And yeah, we're looking forward to see you at the next event. I want to wish everybody a happy rest of the week and hope to see you at the next event. Thank you, everyone. Bye now.